Welcome to AP Daily Video, Topic 7.1, Introduction to Natural Selection. My name is Tanea Hibbler, and I teach at Brophy College Preparatory here in Phoenix, Arizona. I'm here to let you know that you can do this. What will we learn today? So, what is natural selection? What factors influence natural selection? And how does natural selection affect populations? Natural selection is a major mechanism of evolution. It's what drives evolution. Evolution is a change in the genetic makeup of a population over time, and it's supported by multiple lines of evidence. Natural selection is a process by which organisms having adaptations that are suited for a particular environment have a greater chance of survival and reproduction, thereby passing those adaptations on to subsequent generations. Charles Darwin, here to the right, is mainly credited with the development of the theory of natural selection. There are several conditions that must be met for evolution by natural selection, so we'll talk about a few of those today. First, there's competition for limited resources. Not all organisms in a population have the same chance for survival. Competition is when organisms are struggling with other organisms to get limited resources. Organisms will compete for resources like space, food, mates, nutrients, and light. Differences in the phenotypes of these organisms will determine how competitive the organism is. This can lead to differential survival. So there's more born than survive. Favorable phenotypes can improve chances of survival. And there's typically variation in a population. Variation refers to genetic differences among organisms within a population, as we see to the right. Not all the individuals look the same. Mutations and sexual reproduction can increase variations within populations. Adaptations are the traits that provide an advantage in a particular environment. And individuals with adaptations have a greater chance of survival and reproduction. The number of individuals within a population having adaptations can increase over time. Evolutionary fitness is measured by reproductive success. So fitness refers to the ability of an organism to survive and produce fertile, viable offspring. Reproductive success refers to the production of offspring. And reproductive success over several generations is a component of evolutionary fitness. Heritability is the ability to pass on adaptations to successive generations. Due to selection, the traits of individuals that have more reproductive success become more common in the population. The reproductive success and the heritability of adaptations contribute to evolutionary fitness. Ecosystem stability determines the rate and the direction of evolution. There are biotic and abiotic environments. They can remain more or less stable. Environments can experience major disruptions and they can change quickly or they can change slowly over time. And I have examples here to the right. So populations are less likely to evolve in environments that remain stable over a long period of time. So in this stable ecosystem, you can see the environment's not changing, and as a result, the population's not changing either. On the bottom, though, we have a more unstable environment. So the more unstable, the faster the rate of evolution. So here you can see this fast changing ecosystem. The habitat has changed, and as a result, the population has also changed. Let's look at an example. Evolution leads to quiet crickets in Kauai. So in this population, there was variation. The males have specially shaped wings. These male crickets have specially shaped wings with a scraper and teeth that produce a chirp when they're rubbed together. But due to a random mutation, some male crickets in the population have a mutation in their wings, and so they're silent like the females in the population. Both of these traits are heritable, so they can be passed on to successive generations. And there's competition that's happening within the population as well. Males are competing for mates, and chirping males traditionally have had more reproductive success. And then biotic factors. So what happened in this particular environment to change the population? On one of the islands in Hawaii, on Kauai, there was an invasive fly species that lays maggots on the back of the crickets, parasitoid maggots. The maggots burrow into the crickets and they emerge a week later, killing the cricket in the process. The flies use a sound to locate the crickets. So, as a result of natural selection, the silent male crickets are more likely to survive to reproduce because the flies can't find them to lay eggs on. So over time, this population became more silent. Silent males now make up 96% of the population of males. It's a great example. Let's go ahead and practice. The lesson skill focus is visual representation. Describe characteristics of a biological concept, process, or model represented visually. So go ahead and pause your video here. Please make sure you look at the diagram, read the labels, read the question, and then select the best answer. Okay. Did you select answer C? Which of the following statements best describes the change in the tree sparrow population? Tree sparrows with lower body masses were less likely to survive and reproduce after the snowstorm. So this is an example of directional selection. What should we take away from today's lesson? Natural selection is the process whereby organisms with adaptations suited for a particular environment will have a higher chance of survival. Natural selection is influenced by environmental stability, genetic variation, adaptations, and fitness. And natural selection can result in an increase or a decrease of specific phenotypes within a population over time. Thank you so much for joining me today, and remember that you can do this. Welcome to AP Daily Video, Topic 7.2, Natural Selection. My name is Tanea Hibbler, and I work at Brophy College Preparatory here in Phoenix, Arizona. And I'm here to let you know that you can do this. What will we learn? So why is phenotypic variation important to a population? How does natural selection act on phenotypic variation? How do changing environments apply selective pressures to populations? And what variables increase or decrease fitness of an organism? Natural selection acts on phenotypic variations in populations. Genetic variation describes the genotypic and the phenotypic differences between individuals in a population. Genetic variation can lead to different phenotypes and or adaptations. Due to natural selection, individuals with more favorable traits are more likely to survive and to reproduce. Genetic variation can increase the probability a population of organisms will survive under changing environmental conditions. So if the environment changes, one of the phenotypes may be better suited for the changed condition. So variation is good. Environments change and apply selective pressures to populations. Selective pressure refers to any biotic or abiotic factors influencing survivability. For example, disease, predation, climate, and food availability. So you can have an organism that has an adaptation and it can be selected for, and an organism without that can be selected against. And the changing environments introduce different selective pressures on populations. And as you see here, individuals' fitness is relative to the environmental condition. So if the environment changes, who has the adaptation or what's considered adaptation can change. So now after 2 years, the organism that originally had the adaptation is now being selected against, and the individual with the other adaptation or the other trait is now being selected for. So the phenotype selected for can be selected against when environmental conditions change and vice versa. Let's look at an example. Environments change and apply selective pressures to populations. In England, peppered moths vary in color. As you see to the right, some are lighter colored and some are darker colored. After the Industrial Revolution, soot from coal burning factories and homes covered many of the trees. As a result, the darker colored moths were well camouflaged and more likely to survive to reproduce. Over time, the dark colored phenotype became most common within this population of moths. But after the Clean Air Act was passed and pollution was cleaned up, the lighter phenotype became more common once again. Some phenotypic variations significantly can increase or decrease fitness of the organism. And fitness refers to the reproductive success over generations. Any phenotype that decreases chances of survival and reproduction negatively impacts fitness. Any phenotype that increases chances of survival and reproduction positively impacts fitness. Let's look at an example. So DDT resistance significantly increases fitness of insect populations. DDT was a commonly used insecticide to combat insect-borne diseases, but it was later banned in the US in 1972. DDT is still used, though, however, in some countries to combat malaria, which spreads through mosquitoes. Due to natural variation in populations, some individual insects are resistant to DDT. As you can see in the picture above, to the right, you have the red insect. It has a natural resistance. There's variation in the population, and that individual is resistant. Resistant individuals are not killed by the DDT, and they survive to pass on this resistance to their offspring. So over time, an entire population of insects can become resistant since those individuals survive to pass on their traits. And with DDT, this can happen in from 2 years to up to 20 years. What should we take away from today's lesson? Phenotypic variation increases the probability a population will continue in unstable environments. Natural selection acts on phenotypic variation by selecting individuals with the most advantageous traits based on certain environmental conditions. Changes in environmental conditions can change which individuals in the population will be selected for or selected against. And any phenotype that increases an organism's chance of survival and reproduction will significantly increase an organism's fitness and ultimately increase the fitness of the entire population. Thank you so much for joining us for this AP Daily video. My name is Tanea Hibbler, and remember, you can do this.
Welcome to AP Daily. This is Mr. Burris from Union High School in Vancouver, Washington. Today's topic is 7.2. So what will we learn today? In today's lesson, we're going to look at what does fitness refer to? How does molecular variation within cells provide organisms with increased ability to respond to a variety of environmental stimuli? And finally, how does molecular variation within cells provide organisms with increased fitness? So let's dive in. Variation can be evident on a cellular and molecular level. It includes differences in molecular structure, differences in molecular types, proteins, carbohydrates, lipids, etc. Differences in the number of molecules present. Variation increases fitness. Individual fitness refers to an individual organism's being able to survive and reproduce. Individual fitness contributes to a species fitness. Not every individual within a species need to show fitness for the species to continue generationally. The more variation within individual organisms in a population, the better chance a species can demonstrate fitness generationally under changing environmental conditions. So let's take a look at an example. This example is being used to illustrate the concept of molecular variation. Chlorophyll molecules vary within plant cells. Different types of chlorophyll capture light energy at different wavelengths. On the screen, you'll notice we have chlorophyll A, chlorophyll B, chlorophyll C. Each one has a different molecular structure. Greater flexibility to absorb incoming wavelengths of light is used for photosynthesis for different times of day and different times of year. Here's another example to illustrate the concept of molecular variation. The cell membrane of animal cells contain a special lipid called cholesterol, while plant cell membranes do not contain cholesterol. Cholesterol helps regulate membrane fluidity. It stabilizes the membrane structure at high temperature, and it maintains fluidity at lower temperature. Properly structured membranes are critical for cell homeostasis. It can also decrease water penetration. So unlike plant cells, animal cells don't have cell walls to help counter lysis. Increasing cholesterol can restrict diffusion of water through the bilayer. So let's take a look at one more example to illustrate the concept of molecular variation. Hemoglobin is a molecule that carries oxygen in blood. There are two types of hemoglobin found in humans. There's fetal hemoglobin, or hemoglobin F, which enables efficient transfer of oxygen from the blood of the mother to the developing fetus. The other type is adult hemoglobin, or hemoglobin F, which binds to oxygen brought in by the lungs and aids in oxygen delivery to body cells. So let's practice. In today's practice, the skill focus is argumentation. You're going to provide reasoning to justify a claim by connecting evidence to biological theories. So what I would like you to do is pause your video and then read the prompt. Look for the boldface word, which is the task verb, and then ask yourself, what are you being asked to do? Then reread the prompt and come up with your answer. When you have your answer, please push play again. Welcome back. For this question, you were asked to provide the best reasoning for the researcher's claim that new species will have a higher reproductive rate compared to Euglena, having only two types of chlorophyll molecules. So your answer should have been A. The additional chlorophyll allows the new species to have faster photosynthetic rates, leading to more photosynthetic products that can be used for metabolic processes like reproduction. The addition of chlorophyll leads to variation in the number and types of chlorophyll, which leads to a fitness advantage for the organism. So what should you take away from today's lesson? In today's lesson, fitness on an organism level refers to the ability to survive and reproduce. Variation at the molecular level provides organisms with the ability to respond to a variety of environmental stimuli. And finally, variation at the molecular level can provide organisms with fitness advantages under changing environmental conditions. Well, I hope this video was helpful. Until next time, thank you.
Welcome to AP Daily, topic 7.3, artificial selection. My name is Tanea Hibbler. I teach at Brophy College Preparatory in Phoenix, Arizona, and I'm here to let you know that you can do this. So what will we learn today? How can humans affect genetic diversity within a population? So through artificial selection, humans affect variation in other species. Artificial selection is a process by which humans select desirable traits in other species and selectively breed individuals with the desired traits. Commonly used in agriculture and economic practices. For example, growing certain crops with specific characteristics or breeding certain animals for sport or sale. This may result in phenotypes that would not otherwise exist in nature, and it can lead to more genetic diversity or less genetic diversity. Let's look at an example. Through artificial selection, humans are affecting variation in other species. So we have a population here, and there's variation within the population prior to artificial selection. Humans can select any trait or combination of traits as desirable and choose to breed individuals within the population to get that desired outcome. Depending on which traits are selected for and how often they are selected, the genetic diversity of the population can change over time. So take a look at the traits that you see in the population to the right, and now selection is going to happen, but it's going to happen with the humans choosing, and it looks like they've chosen particular colors and particular sizes. So this is what the population now looks like after artificial selection. What should we take away from the lesson today? Humans affect diversity within a population by breeding plants and animals and selecting for desirable traits. Thank you so much for joining me today for this AP Daily video. My name is Tanea Hibbler, and remember, you can do this.
Good morning, good afternoon, AP Bio. This is Mr. Mansour coming to you from Tiffin Columbian High School, and welcome to topic 7.3, artificial selection. In daily video 2, this lesson will explain how humans, like yourself, can affect diversity within a population. So what are we going to learn? Always the most important thing. Well, what we're going to learn is what is artificial selection, and how do humans use artificial selection to affect genetic diversity within a population. These are two things that will pop up throughout the Unit 7 course, is this idea of selection. In this case, we're focusing on artificial selection. So through artificial selection, humans affect variation in other species. I have a picture of dogs. I have one here. You might have one at home. There may be one sitting watching this video with you. But artificial selection is the process by which humans select desirable traits in other species and selectively breed individuals with a desirable trait. So this idea that we as humans, we select these traits that are going to benefit us in some way. Commonly used in agriculture and economic practices, for example, growing certain crops with specific characteristics or breeding certain animals for sport or sale. So these are all examples of artificial selection. Artificial selection is important to understand that it may result in phenotypes that would not otherwise exist in nature. And so that's why you've probably already learned about natural selection. Now we're talking about artificial selection and why sometimes we see these phenotypes that would otherwise not exist. With artificial selection, it can lead to more genetic diversity or even less genetic diversity, depending on what trait is being selected for and how long that process has occurred. So let's look at some examples. So like I said, you might have an example sitting right by you. Maybe not quite like this one, but through artificial selection, humans affect variation in other species. Again, that's key. And so hundreds of breeds of dogs have been bred from a single wild species as a result of selecting breeding by humans. So you may have heard of wolves, right? These are the little animals I have sitting on my desk, this and this, very closely related. And so through artificial selection, we have been able to pick these physical and behavioral characteristics that have met our needs. In this way, different breeds of dogs, and I've got all kind of dogs there. You can probably pick yours out on the screen, or maybe if you have one that's not maybe a mutt or whatever. But in this way, different breeds have been suited for different tasks. So I'm just going to kind of have you circle. You can pause it, try to find your dog on the screen. But all of these breeds have been selected for some particular characteristic. So, and these characteristics can be both physical or behavioral. Through artificial selection, we've also, in other species, we talked about animals, let's talk about plants. Everybody's favorite, broccoli and cauliflower. And so different parts of the brassica plant have been developed by human selection to produce at least 6 distinct different vegetables. All 6, I'm probably sure, on your top list. We've got cauliflower, Brussels sprouts, everybody's favorite, kohlrabi, kale, cabbage, and broccoli. I got that question mark in the middle because that shows us that through artificial selection, humans have selected different characteristics, different parts of the plant to come up with these different types of vegetables, which we all love. And remember, eat your broccoli. Through artificial selection, here's another example you might be familiar with. Chickens, right? Chickens were first domesticated in Southeast Asia about 8,000 years ago from the red jungle fowl ancestor. And so chickens have been selectively bred to improve meat, egg nutrition, taste, texture, and even size. So here is your, again, the key is selective. We're choosing these specific traits. And so there you go. You have your red jungle fowl ancestor over the course of time, through the course of artificial selection. We now have chickens today, which you think of. So again, just another example you might be familiar with as you're going through the grocery store. So what should we take away? Key takeaways. 1, humans affect diversity within a population by breeding plants and animals and selecting for those desirable traits. And so again, the key is humans are doing the selection. 2, artificial selection is the process by which humans select desirable traits. Again, we can affect the diversity by selecting these particular traits. 3, artificial selection may result in phenotypes that would not otherwise exist in nature. If you think about some of those dogs from that previous slide, some of those may not exist in nature had it not been for humans and artificial selection. And the last one, artificial selection can lead to more genetic diversity or less genetic diversity, just again, depending on the organism, depending on what trait was selected. And so just seeing how humans have impacted natural selection. So hopefully you found this video useful, and I just want to say thank you, and I'll see you in a future AP Daily. Mr. Mansour signing off from beautiful Tiffin Columbian High School.
Welcome to AP Daily. This is Mr. Burris from Union High School in Vancouver, Washington. Today's topic is 7.4, population genetics. So what will we be learning today? For today's lesson, we'll be looking at what factors drive evolution, how do mutations contribute to natural selection, how does genetic drift impact population size, and then finally, how does the reduction of genetic variation impact populations of the same species? So let's dive in. Evolution is driven by random occurrences. A mutation is a random change in an organism's genome. It's an alteration in a DNA sequence. It can contribute to changes in the genetic makeup of a population over time. It creates genetic variation within a population and provides new phenotypes that contribute to evolution by natural selection. Reduction of genetic variation can increase the differences between populations of the same species. Genetic drift refers to the random change in the frequency of a particular allele within a population. It is a non-selective process generally occurring in small populations. There's an increased death rates and low reproductive rates. Natural catastrophes, such as fires and volcanic eruptions, can cause this small population. Bottleneck events can contribute to genetic drift. A large, diverse population suddenly is reduced to a small population. So in the picture, you can see a bottleneck event occurs, and what happens is a size of the population is severely reduced. Like stated before, genetic drift is a non-selective process occurring in small populations. Founder effect refers to a random process that reduces genetic variation within a small population due to separation from a larger population. Migration and geological events can isolate populations. The genetic makeup of a founder population can be different from the original population. So as shown in the picture, a small group of individuals breaks off from a larger population and establishes a colony. Migration or gene flow is the movement of individuals between populations causing an exchange of alleles between populations. It introduces new genes into populations, resulting in increases in genetic variation. Continued migration between populations reduces genetic diversity between populations over time. So random processes play a role in the evolution of specific populations. Genetic variation is the raw material of evolution. Fitness is relative to specific environmental conditions. So as conditions change, fitness can change. Different phenotypes can be selected for or against according to changes in the environment. And then finally, evolution of a population cannot occur if there is no genetic variation within the population. So let's practice. For today's practice, we're going to focus on stating the null and alternative hypothesis or predicting the results of an experiment. A null hypothesis is one in which states experimental variables have no relationship and experimental observations are the result of chance. An alternative hypothesis is one of several hypotheses stating experimental variables have a relationship and the experimental observations are a result of some non-random cause. So let's practice. Please pause your video, read the question, and formulate your answer. When you're ready, push play again. Welcome back. For this question, you were looking at which of the following is a valid null hypothesis for this investigation involving northern spotted owls. Your answer should have been B, spotted owls will continue to nest in the area where the barred owl songs are being played. So what should you take away from this lesson? For this lesson, number one, evolution is driven by random occurrences. Next, mutation is a random process that results in genetic variation, which provides phenotypes on which natural selection acts and contributes to evolution. 3, genetic drift is a non-selective process occurring in small populations through bottleneck and founder effects. And finally, the reduction of genetic variation within a given population can increase the differences between populations of the same species. Well, I hope this video has been helpful. Until next time, thank you. Welcome to AP Daily. This is Mr. Burris from Union High School in Vancouver, Washington. Today's topic is 7.5, Hardy-Weinberg equilibrium. So what will we be learning today? For today's lesson, we're going to look at what is the Hardy-Weinberg model, what are the conditions under which allele and genotype frequencies will change in populations, how are allele and genotype frequencies calculated using the Hardy-Weinberg equations, what do changes in allele frequencies provide evidence for, and then finally, what are the impacts on the population if any of the conditions of Hardy-Weinberg are not met? So Hardy-Weinberg is a model for describing and predicting allele frequencies in a non-evolving population. A population in Hardy-Weinberg equilibrium is not evolving. Frequencies of alleles and genotypes stay the same generation after generation for a population in Hardy-Weinberg equilibrium. 5 conditions that must be met for a population to be in Hardy-Weinberg equilibrium include 1, a large population, meaning no genetic drift. 2, an absence of migration, so no gene flow. 3, no net mutations, so no genes are modified, deleted, or duplicated. 4, random mating, so no sexual selection. And then finally, an absence of selection, so no natural selection. These conditions are rarely met, but they provide a valuable null hypothesis. Allele frequencies in a population can be calculated from genotype frequencies using the Hardy-Weinberg equations. P squared plus 2pq plus q squared equals 1 is one of those equations, where p squared is equal to the frequency of the homozygous dominant genotype, 2pq is equal to the frequency of the heterozygous genotype, and q squared is equal to the frequency of the homozygous recessive genotype. Remember, this is used to determine genotype and or phenotype frequencies of individuals in a population. P plus Q equals 1 is where P is equal to the frequency of the dominant allele and Q is equal to the frequency of the recessive allele. This is used to determine the frequency of a particular allele in a population. Now, changes in allele frequencies provide evidence for the occurrence of evolution in a population. These factors can disrupt Hardy-Weinberg equilibrium and change allele frequencies within a population. One of these factors, mutations. Changes in genes can occur through random events that delete, insert, or substitute nucleotides. Another factor, non-random mating. Individuals choose to mate with another based on certain traits. Gene flow is another factor where new genes can be introduced to populations. So migration of individuals in and or out of a population. Genetic drift is where changes in the allele frequency within a population can occur due to random environmental events. Think bottleneck founder effects. And finally, natural selection. Alleles improve or reduce fitness for individuals to survive and reproduce in a given environment. So let's look at an example. We're going to look at a population of beetles in Hardy-Weinberg equilibrium. The population consists of black and red beetles. The dominant allele is black, and we'll use capital A for that allele. And the recessive allele is red, and we'll use lowercase a. The black beetle population consists of the homozygous dominant and heterozygous genotypes, and the red beetle population consists of the homozygous recessive genotypes. For this population, the dominant allele, P, is equal to 0.3, and the recessive allele, Q, is equal to 0.7, with P plus Q equals 1, so 0.3 plus 0.7 is equal to 1. Now we're going to look at what is the frequency of the homozygous dominant genotype. Well, we're going to use the equation P squared plus 2PQ plus Q squared equals 1. P squared is the frequency of the homozygous dominant genotype. So P squared is equal to 0.3 squared equaling 0.09 or 9%. For the heterozygous genotype, it is equal to 2PQ, which is equal to 2 times 0.3 times 0.7, which equals 0.42 or 42%. And then finally, what is the homozygous recessive genotype? Well, that is equal to Q squared, which is 0.7 squared equaling 0.49 or 49%. It is important to be mindful of your decimals when you're working with the Hardy-Weinberg equation. So now let's look at an example that you're going to work through. For this problem, I would like you to pause your video, solve the problem, and then push play when you're ready with your answers. Welcome back. For this question, you were looking at what is the frequency of each genotype in this population and what is the frequency of the dominant phenotype. The frequency of the homozygous dominant genotype is 0.36. The frequency of the heterozygous is 0.48. And the frequency of the homozygous recessive is 0.16. Now, what is the frequency of the dominant phenotype? Well, that is going to be equal to both P squared and 2PQ because both of them are going to exhibit the dominant phenotype. And the answer there would have been 0.84. So now we're going to practice some more. For this practice, you're going to perform a mathematical calculation, including mathematical equations in the curriculum. So for this practice problem, I would like you to hit pause, read the question, formulate your answer, and then hit play again. Welcome back. For this question, you were supposed to look at, based on the information provided, which of the following best represents the calculated differences between the isolated population and the general population. Your answer should have been D, that the frequency of the Ellis-Van Creveld allele is 0.0447 in the isolated population and 0.0026 in the general population, which suggests that genetic drift has occurred in the isolated population. A couple key points on solving this question is that Q squared was given to you, where in the general population, it was 1 in 150,000 live births. And then in the isolated population, it was 1 in 500. And the genetic drift occurred due to those isolated populations. So what should you take away from this lesson today? First, Hardy-Weinberg is a model for describing and predicting allele frequencies in a non-evolving population. 2, the conditions for a population to be in Hardy-Weinberg equilibrium are a large population size, absence of migration, no net mutations, random mating, and absence of selection. The allele and genotype frequencies can be calculated using the Hardy-Weinberg equation, where P and Q represents the alleles, and P squared, 2PQ, and Q squared represent the genotypes. Changes in allele frequencies provide evidence for the occurrence of evolution in a population. And then finally, a population can evolve if the conditions of Hardy-Weinberg are not met, with small populations being more susceptible to random environmental impacts than large populations. Well, I hope this video has been helpful. Until next time, thank you. Welcome to AP Daily. This is Mr. Burris from Union High School in Vancouver, Washington. Today's topic is 7.6, Evidence for Evolution. So what will we be learning today? For today's lesson, we're going to look at what evidence supports evolution, how are fossils dated, how do morphological homologies provide evidence for common ancestry, how do fundamental molecular and cellular features shared across all domains of life provide evidence of common ancestry, and then finally, what evidence supports relatedness of organisms in all domains. So let's dive in. Evolution is supported by scientific evidence. Molecular, morphological, and genetic evidence from extant and extinct organisms adds to our understanding of evolution. Evidence supporting evolution includes the following. Geographical. This is evidence based on characteristics of a habitat or land area. There is geological, which is evidence based on environmental features of the Earth over time. Fossils document patterns of evolution and changes in environmental conditions over time. There's physical, which is evidence based on phenotypes of species. There's biochemical, which is evidence based on chemical composition of living things. So basically, comparison of biomolecules such as DNA and proteins. And then finally, mathematical evidence based on calculations and statistics. Mathematical models and simulations are used to illustrate and support evolution. Fossils are the preserved remains, impression, or trace of once living organisms from the past. Fossils can be dated using a variety of methods. You can use the age of rocks where a fossil is found, the rate of decay of isotopes, including carbon-14, and geographical data. While fossil records are incomplete, they provide information about Earth's history and the living world that existed at that time. Morphological homologies represent modified traits shared among different species. Homologous structures are a variation in a structure that was present in a common ancestor. For example, the bone structure of human arms, cat legs, horse legs, bat wings, and dolphin fins, as you can see on the screen. Vestigial structures have a reduced or obsolete feature that serve little or no purpose for the organism. Example, the bone structure for legs in some snakes, the hip bone structures in whales, and the human tailbone. Analogous structures are structures that evolved independently in different species due to similar environmental conditions, selective pressures. Biochemical evidence is a comparison of DNA nucleotide sequences and or protein amino acid sequences. This provides evidence since all living organisms share the same or highly similar genetic code, the same or highly similar process of gene expression, and the same genetic language. The table on the bottom here is about the divergence in percent of mitochondrial DNA sequences among 5 primates. The more amino acid differences among the 5 different mammals, the more time that has passed since their divergence. The human and chimpanzee diverged more recently in comparison to the human and gibbon based on the mitochondria differences. Many fundamental molecular and cellular features and processes are conserved. Structural and functional evidence supports the relatedness of organisms in all domains. DNA and RNA are carriers of genetic information through transcription, translation, and replication. Major features of the genetic code are shared by all modern living systems. And finally, the metabolic pathways in order to produce ATP are conserved across all currently recognized domains. For example, glycolysis. So let's do some practice. For today's practice, you're going to be describing the data from a table or graph, including identifying specific data points. For this question, what I would like you to do is pause your video, read the question, analyze the data table, and formulate your answer. When you're ready, push play. Welcome back. For this question, you were looking at what evidence would best account for the difference in the primary nitrogenous waste products of tadpoles and frogs. Your answer should have been B, differences in the biochemical process occurring in frog cells and tadpole cells. So what should you take away from today's lesson? First, molecular, morphological, and genetic evidence from extant and extinct organisms add to the understanding of evolution. Fossils can be dated by a variety of methods to provide evidence for evolution, for example, relative and radioactive dating techniques. Morphological homologies provide evidence of shared common ancestry. Many fundamental molecular and cellular features and processes are conserved across organisms. For example, similar genetic codes and processes that synthesize ATP. And finally, structural and function evidence supports relatedness of organisms in all domains. Well, I hope today's lesson was helpful. Until next time, thank you. Welcome to AP Daily. This is Mr. Burris from Union High School in Vancouver, Washington. Today's topic is 7.7, Common Ancestry. So what will we be learning today? In today's lesson, we're going to look at what structural evidence supports common ancestry of all eukaryotes. So let's dive in. Evidence that supports common ancestry for all eukaryotes includes membrane-bound organs, linear chromosomes, and genes that contain introns. Membrane-bound organelles are structural evidence for common ancestry of all eukaryotes. Chloroplasts and mitochondria have structural similarities. They have a double membrane, they contain circular genomes, they contain ribosomes, and then endosymbiotic theory describes how membrane-bound organelles evolved from previously free-living prokaryotic cells, providing evidence supporting these similarities. Nucleus contains linear chromosomes. It also contains a nucleolus. And finally, there's various other eukaryotic organelles that have similar structure and function, such as the ER, Golgi, lysosomes, and vacuoles. Linear chromosomes are evidence of a common ancestry for all eukaryotes. Eukaryotes contain multiple linear chromosomes. The chromosomes are made up of tightly coiled DNA with histone proteins. They're capped with telomeres. They're found inside the nucleus, and the genomes are large. This differs from prokaryotic genomes. Prokaryotic genomes contain single circular chromosomes. They occur in the cytoplasm, and the genomes are small. The genetic code of all eukaryotes contain introns. Introns do not code for proteins, and RNA processing removes introns prior to protein synthesis. So let's practice. For today's practice, you're going to predict the causes or effects of a change in disruption to one or more components in a biological system based on a visual representation of a biological concept, process, or model. So for today's practice, I want you to pause the video, read your question, analyze the figure, and come up with your answer. When you've done that, push play again. Welcome back. For this question, you were looking at which of them best predict the result of the bacteria failing to uptake the recombinant plasmid. The best answer is C. The bacterial cell will not produce eukaryotic genes. So what should you take from today's lesson? The one key takeaway is membrane-bound organelles, linear chromosomes, and introns provide evidence of common ancestry of all eukaryotes. I hope this lesson has been helpful to you today. Until next time, thank you. Welcome to AP Daily. This is Mr. Burris from Union High School in Vancouver, Washington. Today's topic is 7.8, Continuing Evolution. So what will we be learning today? In today's lesson, we'll look at how has it been determined that all life has evolved and continues to evolve? How does resistance contribute to evolution? And finally, how do pathogens contribute to the evolution of other populations? So let's dive in. Life continues to evolve within a changing environment. Evolution is an ongoing process in all living organisms. Populations of organisms continue to evolve. For example, the finches of the Galapagos Islands. Genomic changes over time lead to all species evolution. The mechanisms of genetic change include changes in DNA, which involves gene mutations, chromosomal mutations, cell division, where sexual reproduction increases gamete diversity, independent assortment, crossing over, and environmental disruptions, sudden changes in the environment, and then changes in allele and/or gene frequencies within a population. Continuous change in the fossil record is evidence of the continued evolution. Fossils are a preservation or impression of an organism that lived in the past. They provide a record of an organism over time. Strata layers arrange fossils in the order they were deposited. New fossils mark changes in the environment. New selective pressures can result in those changes in the frequency of certain adaptations within a given population. Transition fossils can show the evolutionary changes as one group evolves into another. Fossil records offer an incomplete chronicle of evolutionary change. Gaps exist in the fossil record. Resistance to various chemicals has resulted in species evolution. Evolution of resistance to chemicals can become evident through natural selection. Mutations can result in resistance. Resistant organisms could be selected for under conditions where the trait is advantageous. The frequency of resistance can increase within a population over time if the trait continues to provide better fitness. In the picture to the right of the screen, a population of butterflies has pesticide applied. The pesticide-resistant individuals survive to reproduce in generation 1. The butterflies in generation 2 have an increase of resistance to the pesticide, which will continue if the resistant provides better fitness for the butterflies. Evolution of resistance to antibiotics, herbicides, or chemotherapy drugs is an ongoing process. A pathogen is an infectious agent that can produce a disease. Pathogens evolve and cause emergent diseases. Pathogenic genomes experience high mutation rates, which generates increased diversity. Pathogens are chemically compatible with the host, so the pathogens co-evolve with the host, and the presence of pathogens can change the phenotypes selected for and against in the host population. So let's look at an example. Pathogens evolve and cause emergent diseases, as we said earlier. Example, 2009 swine flu pandemic. A new strain of the H1N1 influenza subtype was identified. It was caused by the reassortment of the genes originally found in the human and swine flu. The H protein on the surface of the virus was originally only seen in pigs. Another example is COVID-19's pandemic. It is a variation of the coronavirus family of viruses. First discovered in 2019, primarily it causes respiratory illness. So let's practice. For today's practice, we're going to look at proposing a new next investigation based on an evaluation of the evidence from an experiment. So what I would like you to do is pause your video, read the question, formulate your answer. When you're ready, push play. Welcome back. For this question, you were asked which of the following proposals proposes a new step researchers might take to increase the reliability of the experimental observations. Your answer should have been B. So what should you take away from today's lesson? Today's lesson, first, evidence of the genomic changes within a population over time provides evidence that all life has evolved and continues to evolve. 2, the evolution of resistance to antibiotics, pesticides, herbicides, and chemotherapy drugs leads to a species evolution. And finally, as pathogens evolve, the phenotype selected for and against in host populations can change over time. Well, I hope today's lesson was helpful. Until next time, thank you. Welcome to AP Daily. This is Mr. Burris from Union High School in Vancouver, Washington. Today's topic is 7.9, phylogeny. So what will we be learning today? In today's lesson, we'll take a look at what are phylogenetic trees and cladograms, how are they used to infer evolutionary relationships, and how are they constructed? What is a shared derived characteristic? What is an outgroup, and how is it used to infer evolutionary relationships? What is the most accurate, reliable evidence when constructing phylogenetic trees and cladograms? And how do phylogenetic trees and cladograms illustrate speciation? And why are phylogenetic trees and cladograms constantly being revised? So let's dive in. Phylogenetic trees and cladograms both show relationships between lineages. A phylogenetic tree is a branch diagram showing the evolutionary relationship amongst species. Phylogenetic trees can show changes over time, calibrated by fossils or a molecular clock. A cladogram is a diagram used to show evolutionary relationships amongst species. A clade includes any group on a cladogram sharing a common ancestor. Phylogenetic trees and cladograms represent hypotheses and are constantly being revised based on evidence. Traits that are either gained or lost during evolution can be used to construct phylogenetic trees and cladograms. Shared characteristics are present in more than one lineage. Shared derived characters indicate common ancestry and are informative, as shown on this image. The outgroup represents the lineage that is least closely related to the remainder of the organisms. It provides a reference of comparison that helps determine how the main group fits in the evolutionary tree of life, as shown on the image. Phylogenetic trees and cladograms can indicate speciation has occurred. The node is where two lines meet and represents the most common recent ancestor. The root represents the common ancestor of all species on the phylogenetic tree or cladogram. There are different ways phylogenetic trees and cladograms can be drawn. Diagonal, vertical, or horizontal lines can be used. Orientation can be vertical or horizontal. And finally, a node can be rotated, as shown in the image. Phylogenetic trees and cladograms can be used to infer evolutionary relationships. Construction of phylogenetic trees and cladograms can include morphological similarities from living or fossil species, DNA and protein sequence similarities. Molecular data typically provides more accurate and reliable evidence than morphological traits. A derived character is a trait in a recent species, having evolved from an ancestral trait. These are often presented in a table for easier analysis. The phylogenetic tree and cladogram representing the simple evolutionary relationship is the one used. In the chart to the right and the cladogram, the D polylipus has the most differences in amino acids as they relate to the other organisms. A. forsteria and G. gallus have only 3 differences. E. africanus and E. ferus has only 1 difference in amino acids. There can be free rotation around the nodes on this tree. Let's look at an example. Phylogenetic trees and cladograms represent hypotheses. The hypotheses are constantly being revised based on evidence. The phylogenetic tree number 1 represents the relationship of whales to 6 other mammals. All the organisms shown have a pulley-shaped astragalus bone in the ankle except for the whale. Based on the genomic information in the data table provided, phylogenetic 2 was constructed based on new evidence. That new evidence includes the camel is the outgroup with none of the 13 sequences. The peccary and pig have the fewest sequences but are similar. The deer and cow share the same half of the 13 sequences. And the whale and hippo have a similar pattern of DNA sequences. So let's look at an example. For this example, I would like you to pause your video, construct a cladogram using the template on the screen, and then circle the position on the cladogram that best represents the outgroup. When you're ready, hit play again, and we'll go over it. Welcome back. The correct illustration of the evolutionary relationship between the 4 bear species is shown on the cladogram. The polar bear has 7 amino acid differences compared to the black bear. The polar bear has 8 amino acid differences with the brown bear. The black bear and brown bear only have 1 amino acid difference. The panda is circled as the outgroup and being the least closely related to the other species. So let's practice. For today's practice, you're going to represent relationships within biological molecules, including flowcharts. For today's practice here, I would like you to pause your video, and then you're going to construct a cladogram using the template on the screen, and then also circulate the location on the cladogram that best represents the most recent common ancestor. Hit play when you have that done. Welcome back. The correct illustration of the evolutionary relationship between the 5 different human populations is shown on the cladogram. There can be a free rotation around the nodes on the tree except for the Lua population. The common ancestor is circled on the cladogram. So what should you take away from today's lesson? First, phylogenetic trees and cladograms are branching diagrams showing evolutionary relationship among species. Phylogenetic trees and cladograms both show relationships between lineages, with phylogenetic trees showing the amount of change over time. Traits that are gained or lost during evolution can be used to construct a phylogenetic tree or cladogram. Shared derived characteristics indicate common ancestry. The outgroup is a distantly related group that is used to show how the main group falls in the main tree of evolution. Molecular data typically provides more accurate, reliable evidence than morphological traits when constructing phylogenetic trees and cladograms. Phylogenetic trees and cladograms can be used to illustrate speciation that has occurred. And finally, phylogenetic trees and cladograms represent hypotheses that are constantly being revised based on evidence. Well, I hope this video has been helpful today. Until next time, thank you. Welcome to AP Daily. This is Mr. Burris from Union High School in Vancouver, Washington, and today's topic is 7.10, speciation. So what will we be learning today? For today's lesson, we're going to look at when does speciation occur? What is the biological species concept? What is punctuated equilibrium and gradualism? What is divergent evolution? What is allopatric and sympatric speciation? And finally, how do prezygotic and postzygotic mechanisms impact speciation? So let's dive in. Speciation occurs when populations are reproductively isolated from each other. The biological species concept provides a commonly used definition of species. It states that species can be defined as a group capable of interbreeding and exchanging genetic information to produce viable, fertile offspring. There are several mechanisms that can separate members of the same population, potentially leading to speciation. Speciation refers to the creation of a new species, and speciation results in diversity of life forms. Reproductive isolation is critical for speciation. Biological barriers keep members of 2 species from 1, interbreeding, and 2, producing fertile offspring. Reproductive isolation prevents gene flow between populations. 2 types of barriers, prezygotic barriers and postzygotic barriers. So what are the prezygotic barriers that prevent production of a fertilized egg? First, habitat isolation. It's where species occupy different habitats and rarely come in contact. Another one, temporal isolation. Species breed during different times of day, seasons, or years. With behavioral isolation, species have different courtship behaviors or mate preferences. With mechanical isolation, reproductive structural differences prevent successful mating and reproduction. And finally, with gamete isolation, sperm of one species may not be able to fertilize the eggs of another species. Now, with postzygotic barriers, they prevent a zygote from developing into a viable fertile offspring. One of those barriers is hybrid inviability. Mating results in a zygote, but incompatibility may stop the development of the zygote. With hybrid sterility, a hybrid offspring is produced that is vigorous, but may be sterile, such as the mule. Finally, with hybrid breakdown, the first generation of hybrids are viable and fertile, but resulting generations are feeble and sterile. Example, certain rice hybrids. Speciation results in a diversity of life forms. Allopatric speciation refers to the evolution of new species due to individuals from the same population being geographically isolated over a long period of time. No gene flow occurs, and separation may expose populations to different selection pressures. With sympatric speciation, it refers to the evolution of a new species due to individuals being reproductively isolated from a surviving ancestral population. There is no geographical barrier. It can result from genetic mutations, such as polyploidy. It can result from habitat differentiation, and it can result from sexual selection. Different ecological conditions impact the rate of evolution and speciation. There are two models used to represent the rate of evolution. First, punctuated equilibrium. With punctuated equilibrium, evolution occurs rapidly after a long period of stasis. What is stasis? Stasis refers to periods of little to no change. Species may experience no change for long periods of time. Changing ecological conditions are the stimulus for evolution. With gradualism is when evolution occurs slowly over hundreds of thousands or millions of years. Ecological conditions change gradually over a long period of time. Divergent evolution occurs when adaptation to new habitats results in phenotypic diversification. Speciation rates can be especially rapid during times of adaptive radiation as new habitats become available. Adaptive radiation refers to the evolution of new species that allows empty ecological roles or niches to be filled. Darwin's finches located on the Galapagos Islands represent an example of adaptive radiation. Changes in the size and form of the beak have enabled different species of finches to utilize different food sources, as shown in the image. So let's practice. For today's practice, you're going to explain relationships, different characteristics of biological concepts, processes, or models represent visually in theoretical contexts. So for today's practice, I want you to pause the video, read the question, analyze the figures, and formulate your answer. Push play when you're ready. Welcome back. For this question, you're going to look at the experimental results that are the most consistent with which of the stages that are depicted in the model. Your answer should be B, the intermediate stage, which is figure 2. So what should you take away from today's lesson? First, speciation may occur when two populations become reproductively isolated from each other. 2, the biological species concept states that species can be defined as a group capable of interbreeding and exchanging genetic information to produce viable, fertile offspring. Punctuated equilibrium is when evolution occurs after a long period of stasis. Gradualism is when evolution occurs slowly over hundreds of thousands or millions of years. Divergent evolution occurs when adaptations to new habitats result in phenotypic diversification, with the possibility of speciation rates being rapid. Speciation may be sympatric, which occurs without geographic isolation, or allopatric, which occurs with geographic isolation, but both result in the diversity of life forms. Prezygotic and postzygotic mechanisms can maintain reproductive isolation and prevent gene flow between populations. This leads to speciation. Well, I hope this video has been helpful today. Until next time, thank you.
Welcome to AP Daily. My name is Tanea Hibbler. I teach at Brophy College Preparatory in Phoenix, Arizona, and I'm here to let you know that you can do this. So what will we learn today? What is convergent evolution, and how does convergent evolution occur? Similar selective pressures result in similar phenotypic adaptations. Convergent evolution is a process by which similar environmental conditions select for similar traits in different populations or different species over time. Similar traits can be observed in distantly related or completely unrelated species. Such traits are referred to as analogous structures. For example, the selective pressure of similar aquatic environments, like what we see to the right, can select for streamlined bodies and vertebrates like fish and mammals. What should we take away from the lesson today? Convergent evolution is a process by which similar environmental conditions select for similar traits in different populations or different species over time. And lastly, environmental changes can lead to similar evolutionary changes across different populations or species due to similar selective pressures. Thank you so much for joining me today for this AP Daily video. My name is Tanea Hibbler, and remember, you can do this. Welcome to AP Daily lesson topic 7.11, variations in populations. My name is Tanea Hibbler. I work at Brophy College Preparatory in Phoenix, Arizona. I'm here to let you know that you can do this. What will we learn? So how does genetic diversity of a species or population affect its ability to withstand environmental pressures? What makes genetically diverse populations resilient? And what determines if an allele is deleterious or adaptive? The level of variation in a population affects population dynamics. Variation is referring to the different combination of alleles or genotypes and phenotypes found in a population. Results from random mutations and crossing over during meiosis, and genotypes in combination with environmental factors determine the individual's phenotype. So genetic diversity influences a population's ability to respond to changes in the environment. Populations with little genetic diversity, they're going to be at risk of extinction. Let's look at an example here. The California condors have little genetic diversity. In 1987, they became extinct in the wild. They've been threatened by habitat loss, poaching, and also lead poisoning. So let's say that we're going to pretend that this is a genetic diversity in historic condor populations. We can see that there are different variants that exist. There's variation within the population. Habitat loss and poaching reduce the condor numbers drastically. The last 27 that were in the wild were captured and they were bred, and they have since been reintroduced into the wild. And so this represents the recovered condor population now with little genetic diversity. Today, the entire population of condors are descendants of 14 of the birds that were bred in captivity. So they now have very little genetic diversity. The existing population is less resilient to environmental change, and it's being maintained now through captive breeding programs. Genetically diverse populations are more resilient to environmental change. Diverse populations have individuals with a variety of adaptations. So let's say this is our environment, and we have individuals that exist within a population. These individuals have variation, as we can see in the image. If the environment now changes, some individuals may not be adapted to that change. They might be selected against. But they're more likely to contain individuals, diverse populations are more likely to contain individuals who can withstand new environmental pressures. And so even though the environment changed, that you have individuals that are going to be doing fine. The environmental pressures can include things like climate change, catastrophic geological events, habitat loss, human interference, change in food source, or predation. Let's look at another example. Not all individuals in a diverse population are susceptible to a disease outbreak. Antibiotic resistance is an example of population resilience as well. Some individuals can withstand exposure to antibiotics, and that's called antibiotic resistance. They pass resistance on to their offspring as well as to other bacteria in the population. So if we look at the image here, we start with the bacteria that's inside of our bodies. Some are drug resistant just naturally. There's variation within the population. And then we are going to take antibiotics to try to cure the infection that we have. And so the antibiotics kill the bacteria that are causing that illness, but they're also going to kill the good bacteria that protects our body from infection. And that resistant bacteria is left behind. So this drug resistant bacteria is not able to grow and take over. And what's worse is they can take that resistance and they can pass it on to other bacteria that didn't previously have that resistance. Alleles that are adaptive in one environmental condition may be deleterious in another. These deleterious traits are those that reduce the chance of survival, while adaptive traits are those that increase the chances for survival. Genes and traits are not good or bad. Let's look at the example here to the right. The shaded phenotype is deleterious when the sand is lightly colored. And so the individuals with that trait are going to be selected against. And then we look below, the shaded phenotype is an adaptive trait when sand is darkly colored. So if there's a volcanic eruption that's changed the environment, now the individuals that are lightly colored are being selected against, and the individual that's darker colored is going to be selected for. So the selective pressures of the environment determine whether a trait is an advantage or a disadvantage. Let's practice. The lesson skill focus is argumentation. Provide reasoning to justify a claim by connecting evidence to biological theories. And let's go ahead and practice. Please pause your video here. This is a long problem. Read the whole thing. Look at the figure and table as well. Provide reasoning to justify this claim by connecting evidence with biological theories. And an answer that would be appropriate would be the mussels with the LAP94 allele are more likely to survive in high salinity environments because the allele encodes cellular activities that prevent water loss in marine environments. So what should we take away from today's lesson? Populations with little genetic diversity are at risk of extinction. Genetically diverse populations are more resilient to environmental perturbation because they're more likely to contain individuals who can withstand the environmental pressure. And the selective pressures of the environment determine the individual's chance for survival. Thank you so much for joining me for today's lesson. My name is Tanea Hibbler, and remember, you can do this. Welcome to AP Daily, topic 7.11, variations in populations. My name is Tanea Hibbler, and I work at Brophy College Preparatory in Phoenix, Arizona, and I'm here to let you know that you can do this. What will we learn today? What factors lead to extinction? How do environmental changes increase the risk of extinction? How do speciation and extinction rates impact species diversity? And how does extinction create new niches? So extinctions have occurred throughout Earth's history. Extinction refers to the disappearance of a species such that no future generations are going to naturally populate the Earth. They naturally occur. They're part of the history of life on Earth. They can be ongoing and they can occur on small scale over long periods of time. They can serve as a marker for geologic time as well. Extinction rates can be rapid during times of ecological stress. They can occur on a large scale. They can wipe out large numbers of species at one time. They can be caused by catastrophic changes to an ecosystem, such as solar flares, rising sea levels, volcanic eruptions, or asteroid impacts. So the Earth has experienced several large-scale extinction events. If you look at the picture to the right, we see the species diversity, and it's going to be decreased. It's going to severely decrease. And you can see we're losing species over time. So this is now the species diversity after the large-scale extinction event. Human activity can drive changes in ecosystems that cause extinctions as well. Global human activity can drive catastrophic ecosystem change, and it can include the following: habitat loss, as seen in the picture to the right, climate change, habitat degradation, pollution, poaching, and the snow leopard, the elusive snow leopard, is one of the species on Earth that has been greatly impacted by poaching. Also, invasive species can also cause extinctions. Speciation and extinction rates can determine the amount of diversity that exists in an ecosystem. Speciation is the process by which populations of organisms become reproductively isolated and new species form. High species biodiversity can result from high levels of speciation and low levels of extinction. Low species biodiversity can result from low levels of speciation and high levels of extinction. Both pictures refer to areas that have a high level of biodiversity, and that can be terrestrial or aquatic. And the Earth is able to recover from large-scale extinction events, and that evidence is also in the geologic record. Extinction provides newly available niches that can be exploited by different species. The niche just describes the role an organism plays within its environment, and the organism's role can be that of a producer, a decomposer, scavenger, consumer, etc. When a species goes extinct, it leaves an open niche for another species to occupy. So look at the picture on the right, you see species diversity. We're losing species due to the extinctions. And that's going to open up the new niche. And then that's the new species diversity. And then organisms are able to occupy that open niche and become new species. So this can lead to rapid speciation rates and adaptive radiation. For example, after dinosaurs went extinct, the ancestral mammal group occupied the open niche and they experienced adaptive radiation. Let's practice. The lesson skill focus is questions and methods. State the null and alternative hypotheses or predict the results of an experiment. Go ahead and pause your video here and read figure 1, look at the table, and then read the paragraph. State the null and alternative hypotheses for this investigation. And you want your answer to sound something like this. The null hypothesis would be there is no relationship between the competition for food and mean body mass. The mean body mass will not change when there is competition for food. An alternative hypothesis would be the mean body mass of both lemur populations will be negatively affected by the competition for food. And what should we take away from this lesson? So evolution can be rapid during periods of ecological stress. Extinction rates can be rapid during times of ecological stress. Speciation and extinction rates can increase or decrease species diversity. And lastly, extinction creates open niches for other species to exploit. Thank you so much for joining me for this lesson today. My name is Tanea Hibbler, and you can do this. Welcome to AP Daily Lesson Topic 7.12, Origins of Life on Earth. My name is Tanea Hibbler, and I work at Brophy College Preparatory in Phoenix, Arizona. I'm here to let you know that you can do this. What will we learn today? What scientific evidence provides support for models of the origin of life on Earth? What are the models for the origin of life on Earth? And what is the RNA world hypothesis? So several hypotheses about the origin of life on Earth are supported with scientific evidence. Geological evidence provides support for models of the origin of life on Earth. Earth formed approximately 4.6 billion years ago. The environment was too hostile for life until about 3.9 billion years ago. And the earliest fossil evidence for life dates to about 3.5 billion years ago. So this provides plausible range of dates for when the origin of life could have occurred. There are several models about the origin of life on Earth. Primitive Earth provided inorganic precursors from which organic molecules could have been synthesized. There was a presence of free energy and an absence of a significant quantity of atmospheric oxygen. But there's also a theory that says organic molecules could have been transported to Earth by a meteorite or other celestial event. Chemical experiments show that it's possible to form complex organic molecules from inorganic molecules in the absence of life. Organic molecules called monomers served as the building blocks for the formation of more complex molecules. So this includes amino acids and nucleotides. And remember, amino acids are the building blocks of proteins, and nucleotides are the building blocks of DNA and RNA. The joining of monomers produce polymers with the ability to replicate, store, and transfer information. The RNA world hypothesis proposed that RNA could have been the earliest genetic molecule. Let's practice. The lesson skill focus, questions and methods, state the null and alternative hypotheses, or predict the results from an experiment. So let's do this lesson skill practice. In figure 1, this is a model of two different experiments designed to test the formation of organic monomers. And please take note of what gases are going to be present in this setup. Pause the video if you need a little bit extra time to study the diagram as well. Two groups of students attempted to recreate the primitive atmospheric conditions of early Earth using the apparatus represented in figure 1. Each group ran the experiment with different gas mixtures in the apparatus. Predict which experiment has the greatest probability of generating amino acids and nucleotides. So experiment 1 has the greatest probability of generating amino acids, but neither experiment will generate nucleotides. Let's just take a moment to break this down for a minute. So amino acids, remember, they're the building blocks of proteins, and the monomers of proteins, the amino acids, they're going to have carbon, they're going to have hydrogen, oxygen, and they're going to have nitrogen, sometimes sulfur. So you at least can build most amino acids if you have these molecules here. But in experiment 2, I don't see any nitrogen, so it's not likely that you would build amino acids. Now, when we think about nucleotides, nucleotides are the building blocks of DNA and RNA. Nucleotides are going to be made up of a sugar, a phosphate, and a nitrogen-containing base. So they also need nitrogen, but they need phosphorus. So I don't see, because of that phosphate group and the fact that I don't see any phosphorus in any of these these gases here that were added, it's not likely that we would generate any nucleotides with either experiment. So what should we take away from this lesson? Geological evidence provides support for models of the origin of life on Earth. Models for the origin of life indicate that life was extraterrestrial or early Earth atmospheric conditions were ideal for forming monomers for life. The RNA world hypothesis proposed that RNA could have been the earliest genetic molecule. Thank you so much for joining me today. My name is Tanea Hibbler, and please remember that you can do this.